Bismillahirrahmanirrahim. Dear students, Assalamu alaikum and welcome back to our class of cell biology, genetics and evolution. We were discussing various topics related to genetics. In the previous few videos, we discussed sex determination, how it can be determined that whether the newborn individual will be male or female. That was sex determination, and we discussed various mechanisms of sex determination. So in this particular video, we are going to discuss sex linkage, or what we call it sex-linked inheritance. Next, we are going to make some crosses. That is, we are going to take an example of this sex linkage or sex-linked inheritance, that how traits in this particular inheritance are transferred from parents to offspring, or in other words, what is the pattern of sex-linked inheritance. First of all, the definition of sex-linked inheritance or sex linkage. The inheritance of a trait, and you know what is trait, a phenotype, a character that is transferred from parents to offspring. So the inheritance of a trait that is determined by a gene, what kind of gene, and that particular gene is located on the X or sex chromosome. So this particular inheritance is known as sex-linked inheritance. So the trait or the character which we are studying or which is under consideration, that is determined by a particular gene, and that gene is located on an X chromosome or a sex chromosome, a chromosome that determines the sex of the individual. And this is what we have already seen, if you remember, in our previous videos, the sex determination. So inheritance of that trait, since it is linked with the sex, linked with the chromosomes that determine the sex, and since those genes are carried by that particular chromosome, the X chromosome, so this is sex-linked inheritance. Now this is the word by word definition of that particular sex-linked. So here it is, the sex-linked inheritance. The same thing, inheritance of a trait that is determined by a gene which is located on one of the sex chromosomes. So this is the sex-linked inheritance. Now, sex-linked inheritance was, so to say, discovered by the famous geneticist T.H. Thomas Herbert Morgan way back in 1910. Now that's long ago, about 110 or so, since we are in 2021. So about 111 years, so to say, or at least 110 years ago, he determined this particular pattern of inheritance, which is linked with the sex of the individual. Morgan was making crosses with the fruit fly, Drosophila melanogaster. And this particular fruit fly, which is common, very common, you can cultivate it or you can grow it as well. If you have some fruits, a salad of fruits, so you can get them growing there. So Drosophila melanogaster. This particular fly has red eyes. The normal eye color is red. So it has, or it is a red-eyed fly, the color of the eyes is red. Morgan soon found in his crosses that there was a fly whose eye color was white instead of red. And soon he found out that this white color is a true breeding color. That is, this particular trait is a genetic trait, and this is not due to some any other factor like environmental or some temporary or transient factor. This is a true breeding trait, that is the white-eyed flies remained white-eyed from generation to generation. So to find the pattern of inheritance of this particular white-eyed fly, what he did that he crossed a white-eyed male with a red-eyed female. So these were one of his initial crosses. Male was white-eyed and the female was red-eyed. So this is W for white and R for red. One thing that he noticed, obviously, when he crossed a white-eyed fly with a male with a red-eyed female, in F1 and in F2 he considered the results of those, and then what he did next that he made a reciprocal cross, that in the next few crosses he crossed a female which was white-eyed with a male that was red-eyed. Now what Morgan found, that results of these two crosses were totally different. The result which he obtained in the first cross, in which male was white and the female was red-eyed, he obtained certain results, and don't worry about it, we are going to take a look at it in all the crosses very shortly, just to discuss his initial crosses or what results did he get when he made these crosses. So in his first cross, a white-eyed male was crossed with a red-eyed female. He obtained certain result, whatever it was, we are going to see it shortly. In the next cross, what he did that he took a white-eyed female and crossed it with a red-eyed male. Now he was surprised to see that the results of these two crosses were totally different, and this is contrary to what we have seen so far, or what Mendel found out. If you remember, when we were discussing the Mendelian inheritance, or the trait of those characters which was studied by Mendel, it made no difference that whether the characters was introduced by male or female. So Mendel tried both. He made one parent male and then the other female, and then he made a reciprocal cross, and the results were same. There was no change in the result. But here what Morgan found out, that it was a totally different situation. The result of this particular cross was totally different from this one. That is, the initial cross and this was the reciprocal cross. The results were totally different. So soon it was found out that the difference in these two results, or when changing the sex of the individual, it changed the results as well. This was because the genes for this particular trait, that is the eye color, it was located on one of the X chromosome rather than on what we call them autosomes. So since the gene of the eye color, or the allele of the eye color, it was located on the X chromosome, so that's why the results were different. Also, Morgan found out that when he made his initial cross, a red-eyed female with a white-eyed male, the F1 were all red-eyed. And in the following cross, that is, this was F1, and then in F2, what he found out, 3:1. That is, these were 3 ratio, or the 3/4 of the result, or 3/4 of the flies offspring, they were red-eyed, and 1/4 or 0.25 or 25% were white-eyed. However, there was a certain peculiar thing about this result, that of these red-eyed flies, the ratio was 2:1, 2 female, 1 male, whereas these white-eyed flies, they were all males. So obviously this is a strange result. According to Mendelian inheritance, this all should have been 50-50. of these three quarters, 50% would have been male and 50% female, and similarly these white-eyed flies would have been 50% male and 50% female. The result was totally different, although he obtained 3:1. So what this 3:1 result showed, that these two alleles or these two genes, that is red and white eye, they are simple alleles, in which this red color allele is dominant over white eye. The allele of the white-eyed trait is recessive to this particular the red-eyed. So that was dominant, the red was dominant over white-eyed. And in the same fashion, when he made another cross in which he made the female white-eyed and the male red-eyed, the result was very strange, as in the F1 he obtained 50-50. 50% were white-eyed and 50% were red-eyed. And in the F2 the ratio was this 1:1:1. So one red male, one red female, one white male, and one white female. So this is suppose male and female, and this is male and this is female. So obviously these are totally different result of the reciprocal cross as compared to what we have seen in the Mendel crosses. So let's see that how these results would be obtained and how we can make a cross between these two flies. So we start with the initial cross of the Morgan, in which a white-eyed male was crossed with a red-eyed female. So let's see that how we can do it. So let's find out how was the first cross. So these are parents. So we have a female parent here and we have a male parent here. This is phenotype is red-eyed and its phenotype is white-eyed. Now we are going to draw the two chromosomes, the X, and we already know that the sex determination pattern in Drosophila is similar to humans. So we have both X chromosomes in female, XX, this is female, and X and a Y, this is a male. So we are going to make symbols for these chromosomes. So a simple straight rod, and we are going to make it R. Capital R is the red trait, and X will be a straight rod and Y will be a slightly curved one. So this will be R, and this particular chromosome, which is Y, it does not carry this particular allele for eye color. So there is no allele on the Y chromosome, and this is why all the results are totally different. Okay, so now let's make the cross. So this is a red-eyed female and this is a white-eyed male. Lowercase r, as I've just told you, is for the white color. Since female has both X chromosome carrying the dominant allele, so it's all gametes will carry this dominant red-eyed color allele. So we are going to make only one gamete. However, the gametes of the male, one will be carrying the X chromosome with the recessive allele, and the other will carry the other will carry the Y chromosome. So now let's make the cross. If these two gametes, say this is sperm, when they fertilize, this particular sperm fertilizes this egg, we get R and R. And if this particular sperm fertilizes the egg, so this is the result. So we get a male, and this male is also red-eyed. So this is these are gametes. and this is F1. So this is F1 in which all the individuals are red-eyed. So let's do the selfing of this F1 and let's uh, make the cross. So this is female and this is male. Now the female, the gametes of the female will be 50-50. 50% will carry the red-eyed color allele and the 50% will carry the white-eyed allele. And similarly, the male will also have 50-50 chromosomes. 50% will carry the dominant allele, red-eyed color, and 50% will carry the Y chromosome, which does not carry any eye color allele. So these will be the gametes of both the parents. These are the gametes of the female, the eggs, and they are 50-50. And similarly, male has R, the red eye color allele, and the other chromosome Y will go to another gamete. So this egg can be fertilized by this particular sperm. So we get a female, and as you can see, it is carrying both the dominant alleles, so it will be red-eyed. But this particular egg can also be fertilized by this particular sperm. So we get a male which is also a red-eyed. Similarly, this particular egg can be fertilized by this sperm. So we get a female, and this particular female will be heterozygous, as this is. And finally, this particular egg can also be fertilized by this particular sperm, and what we get is a male, and this particular male will be white-eyed. Now this is a Y chromosome. So as you can see, as I told you in the start of the video, that the F2 result was, although we have 3:1, as you can see, we have 3:1, 3 red-eyed flies, 3 ratio or 75%, and 25% white-eyed flies. However, the 25% white-eyed flies are all male, and of those 75%, 50% are female and 25% are males. Whereas it should have been 50-50 of these three. Say if we have obtained 75 flies and 25 white-eyed and 75 red-eyed, so of this 25 there would have been 12-13 ratio, and similarly in this 75 we would have obtained the 50 red-eyed males and 50% red-eyed females. But the actual result is totally different, that the males, all the white-eyed flies are males, and of the red-eyed flies we have 50% red-eyed females and only 25% red-eyed males. This was his initial cross, or the first cross, although it is following, as far as the one thing you should note, as far as the color or the trait is concerned, this particular result, the F2, now this is F2, this particular result is following the simple Mendelian inheritance, that red color is dominant over white, and we have obtained the result simple monohybrid ratio of 3:1. 3 red-eyed flies and 1 white-eyed flies. As far as this is concerned, this particular result is following the simple Mendelian inheritance. However, if you take the sex into account, when you take the sex into consideration, then this result is not following the typical Mendelian inheritance. As of these 3, we have 2:1, 2 female and one male, and this one is all male, whereas it should have been 50-50. So this was the first class of Th. Morgan. In the next video we are going to see the reciprocal cross, and you are going to find out that this particular trait or character, since it is located on the, or gene of this particular trait is located on the sex chromosomes, so it doesn't follow the regular pattern of inheritance as other traits follow, which are located on the autosomes. So stay tuned, and we are going to see the reciprocal cross in the next video. Thanks for watching, and see you in the next video.